So imagine you are planning a massive permanent move.
Right.
You currently live on this like freezing, snowy mountaintop, but you are moving to the middle of the scorching Sahara Desert.
Quite the transition.
Yeah, exactly. So how do you prepare for that? Do you go out and acquire a whole bunch of new gear, like cooling suits, sand goggles, maybe a camel?
A camel would help.
Right? Or do you take the exact opposite approach? Do you throw away almost everything you own, travel incredibly light, and just sort of endure the brutal transition until you eventually adapt to the heat?
It is the ultimate question of adaptation. And when we look at how life actually colonizes entirely new ecosystems, we see both of those strategies play out in spectacular and sometimes completely destructive ways.
Do you throw away everything and just endure it? I mean, today we are watching that exact scenario play out on a microscopic scale.
Welcome to today's deep dive. We're getting to a fascinating 2026 Nature Communications paper that tracks this bacterial lineage called Limnocylindria.
Yeah, incredibly ancient group, right, belonging to the Chloroflexota phylum. And we are going to track their epic evolutionary journey as they manage to just completely pack up and move from a highly stable life in the dirt to a radically different free-floating life in freshwater lakes.
And we really need to set the stakes right away here, because we aren't just talking about a bacterium finding a slightly different food source or, moving to a wetter patch of dirt.
Yeah.
We are talking about a complete foundational restructuring of their genomic architecture. I mean, the data we're looking at today challenges long-held assumptions about how evolution, natural selection, and genomic decay actually work in free-living bacteria.
Right. It forces us to ask, like, what does survival actually cost? So let's start with the origins. Where did this journey even begin?
It began in the soil. So the ancestral roots of the Limnocylindria class, they lie in two lineages known as the P211E and QHBO01 families.
Okay.
And these are classic, highly successful soil-dwelling bacteria. To give you an idea of just how successful, QHBO01 can reach a peak environmental abundance of up to 30% in some soil microbiomes.
Oh, wow, 30%.
Yeah, they absolutely dominate their environment.
So they're perfectly comfortable. But then this evolutionary crossroads appears, right? The family tree splits into two sister lineages. One is called CSP14, and the other is Limnocylindraceae.
Correct.
They share the exact same ancestor, but when it came time to expand out of the soil and into the water, they took wildly different paths. Let's look at CSP14 first. This is the group that chose the acquire new gear strategy.
Precisely. So CSP14 expanded from the soil into aquatic sediments and eventually out into the open water column by actively acquiring niche-specific genes.
Just grabbing new tools.
Exactly. And crucially, while doing this, they maintained a very large, complex genome, ranging from 2.4 to 3.2 megabase pairs, with a very high GC content of around 68%.
Okay, let's unpack this. From a human perspective, this seems like the most logical way to evolve, right? They basically became a microscopic Swiss Army knife.
That's a good way to look at it, yeah.
Like, the bacteria that stayed in the soil kept their ancestral tools, like those heavy-duty enzymes needed to break down complex plant polysaccharides. But the ones that moved into the sediment, where oxygen is incredibly scarce, they acquired new tools. The data shows they picked up nitrate reductase and nitrate oxidoreductase complexes.
Exactly, yes. And those complexes allow them to breathe without oxygen. And it goes even further than that. The CSP14 representatives that fully transitioned into open freshwater habitats actually picked up genetic loci for flagella and chemotaxis.
Wait, really? They literally acquired the biological machinery to build little propeller-like tails?
They did.
Just so they could swim and navigate the open water?
They did, yeah. They encountered a new environment and bolted on new hardware. And the genomic evidence backs up exactly how they pulled this off. The analyses in the source material show that CSP14 experienced a significant increase in positive or diversifying selection. And this was alongside a relaxation of purifying selection.
Relaxed purifying selection. Okay, translate that from geneticist into plain English for us.
Sure. So purifying selection is basically nature's strict quality control department. It ruthlessly eliminates mutations to keep things exactly as they are.
Okay.
If a mutation breaks a working part, that organism dies.
Yeah.
But when that pressure relaxes, the genome suddenly has the freedom to experiment.
Oh, I see.
Right? The bacteria can accumulate new genes, increase their genetic redundancy, and essentially trial and error their way into new environments without immediately dying off. It gave them the evolutionary breathing room to grow, adapt, and build that bigger tool belt.
Okay, so if I'm tracking the story so far, time for a quick recap. One, our bacteria started as these highly successful, stable residents in the soil. Two, one sister family, CSP14, moved into the water by aggressively adding new genetic tools like swimming tails and new ways to breathe. And three, to pull all of that off, their genomes had to stay large, complex, and highly versatile.
Exactly. They are the ultimate hoarders of useful genetic tools.
It really makes you wonder about your own problem-solving style, right? Like, think about how you solve problems. When you face a massive transition in your life, are you the type to acquire new skills, take classes, and add tools to your arsenal? Or do you simplify and strip away distractions? Because as we are about to see, the sister lineage, Limnocylindraceae, took that minimalist approach to an absolute extreme.
And it is an extreme defined by, well, catastrophic loss.
Here's where it gets really interesting. Limnocylindraceae also made the jump to freshwater lakes. But they didn't act like a Swiss Army knife at all. Instead, they threw away almost half of their entire genome.
It's staggering.
They dropped from that bulky ancestor down to a median of just 1.3 megabase pairs. They even lost the ability to synthesize 6 essential amino acids. They literally just stopped making the fundamental building blocks of life.
It is a radical biological paradox. I mean, how does a life form become more successful in a brand new, challenging environment by spontaneously forgetting how to build its own essential proteins?
Yeah, it makes no sense.
Right. And to understand this, we have to look at the exact mechanisms of this decay. Because this was not a calculated, elegant shedding of weight. It was driven by the catastrophic collapse of a microscopic defense system known as the base excision repair, or BER pathway.
The base excision repair pathway. So this is essentially the spell checker for their DNA, right?
That's a perfect way to look at it. BER is an essential DNA repair mechanism that constantly patrols the genome to correct small base lesions. Basically, chemical damage to the DNA letters.
Okay.
And in Limnocylindraceae, the enzymes that run this spell checker just went missing, leading to highly specific, chemically devastating consequences. Let's look at the 3 major failures.
Let's do it.
First, the loss of alkylation repair. They lost DNA glycosylases with names like TAG, ALK, and MPG.
Whoa. Okay, stop. That is a lot of alphabet soup right there.
Yeah.
For someone without a PhD in genetics, what do TAG, ALK, and MPG actually do, and what happens when they go missing?
Fair enough, yeah. Let's look at the actual mechanism. So DNA is made of 4 bases: guanine, cytosine, adenine, and thymine. G pairs with C, A pairs with T.
Right, the classic pairings.
Exactly. When those specific enzymes go missing, the bacteria cannot repair a specific type of chemical damage called 3-methylguanine, or 3-meg.
Okay, 3 meg.
Normally, during DNA replication, the molecular machinery sees a guanine and pairs it with a cytosine. But 3-meg is a damaged guanine. Because the spell checker is broken, the machinery misreads it. It pairs it with a thymine instead of the cytosine.
Wait, so it puts a T where a C should be.
Right. And then, when that cell divides again, the machinery reads that new thymine and perfectly pairs it with an adenine. The end result: a permanent typo in the DNA where a guanine has been completely replaced by an adenine.
Wow.
We call that a G to A transition.
Okay, I see. It's a cascading error. So what was the second failure point?
The second major loss involves deamination repair. So they lost a suite of enzymes, including UNG, MUG, and other UDG family protein.
And what's their job?
Their job is to deal with cytosine. Cytosine is chemically unstable. It spontaneously deaminates, meaning it loses an adenine group, and chemically degrades into uracil. Now, uracil is a letter that belongs in RNA, not DNA.
Oh, right. So it's like an alien letter in the genetic code.
Exactly. Without those specific enzymes to cut that uracil out and fix the gap, the replication machinery just assumes it's supposed to be there, and it reads uracil as if it were a thymine.
Oh, no.
Yeah, so it inserts an adenine across from it. Again, this creates yet another permanent G to A transition mutation.
So we were just bleeding guanine and cytosine bases, replacing them with adenines and thymines over and over.
Yep.
And the sources mention a third failure point, oxidation.
Yes, oxygen damages DNA constantly. The enzyme MUT normally acts as a sanitizer. It finds and destroys oxidized raw nucleotides, specifically one called 8-oxo-DTTP, before they can be inserted into the DNA.
Okay, so a cleaner.
Right, but without MUT, that damaged oxidized piece gets built into the DNA strand directly opposite an adenine. This forces an AT pair to mutate into a CG pair, which is a structural change called a transversion.
Yikes.
And conversely, they also lost an enzyme called Ni, which cleans up a different type of oxidation. Missing Ni allows a GC pair to turn into a TA transversion.
Okay, let me put this in perspective. Imagine you are typing a massive million-word manuscript.
Right.
Suddenly, your backspace key is ripped off the keyboard and your spell checker is permanently disabled. But you still have to type faster and faster to hit a deadline. Typos start accumulating exponentially. Entire paragraphs become gibberish. This is what happened to Limnocylindraceae. They entered a hypermutator state where the damage was so unchecked that massive chunks of their DNA simply became inactive nonsense and eventually got deleted entirely.
What's fascinating here is that this absolute genomic train wreck was the key to their survival.
Wait, the train wreck saved them.
It did. The hypermutation generated massive amounts of random genetic variation. Yeah. Even though it was eroding their overall metabolic capacity and destroying those 6 essential amino acid pathways, it also allowed them to randomly stumble off the exact genetic tweaks needed to survive the shock of fresh water. And they did it exponentially faster than a normal, healthy mutation rate ever could have.
Wow. A desperate, chaotic sprint for survival. Let me make sure I've got this straight with another quick recap. 1, Limnocylindraceae lost its base excision repair pathway, their spell checkers. 2, without them, unchecked chemical damage from alkylation, deamination, and oxidation caused massive continuous typos in their DNA. And 3, this hypermutator state caused them to lose half their genome and essential survival pathways, but somehow forced a rapid chaotic adaptation to the lake water.
That is the exact mechanism, yeah.
It's incredible. So here's a question for you listening. If losing your defense mechanisms allowed you to change and adapt instantly to a new environment, would that trade-off be worth it? It's a terrifying thought.
It is a profound trade-off. And the scars of that hypermutation aren't just theoretical. They are permanently written into the actual chemical percentages of their remaining DNA.
Right.
Which leads us to a major visual mystery surrounding their GC content.
Because along with their genome shrinking by half, the actual percentage of those guanine and cytosine bases, the GC content we mentioned earlier, dropped drastically, right? From about 68% in their ancestors down to 63.15%.
Exactly. And to understand why that specific percentage drop matters so much, we have to look at how DNA actually codes for proteins. DNA is read by the cell in blocks of 3 letters, which we call codons.
Okay, codons.
Now, many amino acids can be coded by several different combinations of those 3 letters. A change in the 3rd letter of a codon often doesn't change the resulting amino acid at all. We call this a synonymous substitution.
Got it. So it's like spelling the word color as C-O-L-R or C-O-L-O-U-R. The meaning, the actual protein that gets built, is exactly the same, but the letters used to spell it are slightly different.
That is a perfect analogy, yes. And because of the missing DNA repair enzymes we just spent so much time discussing, Limnocylindraceae was experiencing a massive, unstoppable influx of mutations, turning Gs and Cs into As and Ts.
Because of the deamination and alkylation we talked about?
Precisely. Over time, those synonymous substitutions meant the bacteria were still successfully building the exact same essential proteins to survive, but they were now using AT-rich codons to spell them out, rather than GC-rich codons.
And if you look at the data charts in the source material, it paints a really vivid picture. Imagine looking at a wide funnel, or like the neck of a wine bottle on a graph.
Yeah, it's a great visual.
At the bottom, in the ancestral bacteria, you have this wide, varied use of heavy, GC-rich codons. But as you trace the lines up the evolutionary timeline, moving into the hypermutating Limnocylindraceae, the data visually narrows. The GC-rich codons are squeezed out like a bottleneck, entirely replaced by AT-rich codons. It really looks just like a funnel.
And that visual wine bottle shift gets right to the heart of a major ongoing fight in evolutionary biology. For decades, scientists have debated 2 major ideas: the adaptive versus non-adaptive paradigms of genome reduction.
Oh, let's break that debate down. So the adaptive paradigm basically treats nature like a master engineer, right?
Right.
Hypotheses with cool names like streamlining or the Black Queen hypothesis suggest that bacteria shrink their genomes on purpose. The theory is that they actively delete DNA to save energy, conserve resources, and become ultra-efficient machines in environments where nutrients are scarce.
Exactly. It assumes intent and efficiency. In contrast, the non-adaptive paradigm argues that genome shrinkage in free-living bacteria is largely just an accident. It says that it is the result of genetic drift and severe mutational bias, operating entirely independently of any grand master plan for efficiency. It's just decay.
So scientists are basically fighting over whether the bacteria are like Marie Kondoing their DNA to spark joy and efficiency, or if their house is just burning down.
Yeah, exactly.
How do we know which one is happening here?
Well, the data in this deep dive gives a definitive verdict for Limnocylindraceae. It absolutely proves the non-adaptive paradigm.
Oh, really? How so?
The proof lies in the intergenic regions. Those are the empty, non-coding spaces between the genes.
Okay, the blank spaces.
Right. If the bacterium was adaptively, purposely streamlining its entire genome for efficiency, the GC content would drop uniformly everywhere.
Oh, I see.
But the researchers found that the GC content in the intergenic regions remained significantly higher than in the coding regions inside the genes. The drop in GC was happening much faster inside the genes, simply due to the sheer mathematical probability of those synonymous substitutions accumulating.
Wow.
Yeah, it proves the GC drop is driven by the blind mutational bias of the broken DNA repair enzymes, not by an elegant, calculated master plan.
Okay, let me do a quick recap to lock this in. 1, mutational bias aggressively replaced GNC bases with A and T bases without changing the actual proteins. 2, when graphed, this creates that visual funnel or wine bottle shape we talked about. And 3, because of where those mutations happened, it proves their genome reduction was a chaotic accident, validating the non-adaptive paradigm.
It is pure, undirected decay.
We really do love to think of nature as this perfect, calculating engineer. But think about this. How does it change your view of evolution to know that sometimes survival is just a matter of surviving a series of catastrophic genetic accidents?
It definitely forces us to reevaluate what survival of the fittest actually looks like in practice, because it raises a glaring issue. If the DNA changes were largely a blind accident of decay, where is the actual targeted adaptation taking place? I mean, how are they surviving?
Right. So what does this all mean? If their genome is just a shrinking, hypermutated mess, why are they dominating these freshwater lakes?
The answer lies in the proteins. We spend so much time talking about DNA, but DNA actually only makes up about 3% of a bacterial cell's dry weight.
Just 2%.
Just 3%. Proteins make up a massive 53% of their mass.
Oh, wow.
So the real survival of the fittest adaptation in Limnocylindraceae isn't happening in the DNA. It's happening at the proteome level.
Right. Because remember earlier we mentioned that during their hypermutation phase, they accidentally lost the ability to synthesize 6 essential amino acids.
Yes.
Because of that genomic decay, they became partially auxotrophic. They can't make their own food. They now have to scavenge oligopeptides, which are basically just small, broken chains of amino acids directly from the dissolved matter in the lake water just to survive.
And lake water is a highly variable, stressful environment. Nutrients are scarce.
Yeah.
So to survive this severe limitation, natural selection finally stepped in with surgical precision. It intensely favored bacteria that built their cellular proteins using specific amino acids that inherently require less carbon and less nitrogen atoms to construct.
Oh, it's like a construction company whose global supply chain suddenly breaks down.
Exactly.
That they completely lose their high-tech fabrication pathways, but instead of going out of business, they adapt by figuring out how to build the exact same house using cheaper, locally scavenged materials. In this case, low carbon and low nitrogen amino acids.
That is exactly what happens. And this brilliant optimization is written clearly across their entire proteome. When researchers measure the elemental composition of their proteins, Limnocylindraceae utilizes significantly lower levels of carbon and nitrogen per amino acid compared to their soil-dwelling ancestors. They built a cheaper house.
And here is the ultimate proof that this isn't just a random fluke for this one family. The source data looked at completely unrelated families of bacteria that also made the dramatic jump from soil or sediment into freshwater. They found the exact same algorithm of adaptation.
Yes, the Fis fairy lineage, when it moved to freshwater, optimized its entire proteome to require significantly less carbon. And another entirely separate lineage, Methylophalaceae, transitioned to freshwater and optimized its proteome to require far less nitrogen. It proves that this kind of proteome streamlining, building cheaper proteins, is a broad, repeatable, evolutionary response to nutrient limitation when moving to a new habitat.
It's amazing. Let's recap this final segment. 1, while the shrinking of their DNA was just a random, chaotic accident, the changes to their proteins were highly calculated by natural selection. 2, they adapted to nutrient scarcity by building their proteins with cheaper elemental ingredients like carbon and nitrogen. And 3, entirely unrelated bacteria used this exact same strategy when moving to freshwater.
It is the ultimate form of biological accounting.
It makes you think about your own resources. Like, if your primary resources suddenly became scarce, what expensive habits or materials in your own life would you streamline first just to survive?
Well, if we connect this to the bigger picture, this deep dive has shown us 2 entirely valid ways to colonize a new world. We saw CSP14's path, which was expansive, acquisitive, adding new genes to master the environment. And we saw Limnocylindraceae's path, which was chaotic, mutation-driven decay coupled with ruthless protein-level optimization.
Okay.
Both lineages survived the move to the lake.
But there is a massive hidden cost to the second strategy, right?
A profound evolutionary cost, yes. The tragedy of Limnocylindraceae is hyperspecialization. The hypermutation that allowed it to rapidly colonize freshwater also caused irreversible genomic erosion.
Right.
It threw away so much of its foundational genetic material that it has painted itself into an evolutionary corner.
Wow.
It is beautifully, perfectly adapted to its current freshwater niche, but if that lake dries up or the environment drastically shifts again, it simply lacks the genetic flexibility to adapt a second time. It has nothing left to give. It literally can't mutate its way out of a paper bag anymore.
The hypermutator strategy is a brilliant short-term hack that risks long-term extinction. It allows you to sprint into a new environment, but the sheer speed of the sprint completely burns the bridge behind you. And honestly, it makes you look at human technological and social structures a bit differently.
Oh, absolutely.
We are constantly, rapidly stripping away foundational, traditional repair mechanisms, whether those are social safety nets, deliberate analog processes, or even just time to think. Also, we can adapt faster to a hyper-fast digital ecosystem. Are we, like Limnocylindraceae, trading our long-term evolutionary flexibility just for the ability to survive the demands of right now? Think about it. Thank you for joining us on this deep dive, and we highly encourage you to explore the fascinating source material for yourself.